Hi everyone, I am Anna Bernasconi and with my colleague Pietro Pinoli we are going to present our tutorial Conceptual Models and Databases for Searching the Genome. We both work at the Department of Electronics, Information and Bioengineering at Politecnico di Milano. And let me present you. We are postdoctoral researcher and research fellow at Politecnico di Milano. My main research area is on data modeling, data integration, knowledge management for genomic and biological data. Pietro is in charge of computational biology, data management, big data, and machine learning. Here you find our contacts. And let me say that this tutorial will be divided in 4 parts that we will present in alternation. And there will be also a 5th part which is not shown during the tutorial but is provided as additional material. So our work has been followed during the ERC Advanced Grant from 2016 to 2021 by our principal investigator Stefano Ceri. Its focus has been on data modeling, integration, extraction, search for tertiary genomic analysis. The approach has been a radical change in data management abstractions, and the results it provided are GMQL, a big data management system, an integrated repository called Metabase, a search system, GenoSurf, demonstrated through many biological and clinical research applications. Some of these are going to be seen during this tutorial. We also had a project about the EIT Digital Data Against COVID-19 program. Our innovation activity was called VirusSurf. This has been a creation of an environment that allows customers to submit raw viral sequence data and receive full report regarding many relevant aspects. This has been a collaboration with the TU Delft and the Quantia Consulting Company. So let me guide you through the tutorial objectives. From here, we would like you to learn first how to use conceptual models to clarify the complex domain of genomics. Second, how to represent genomic information in a usable and accessible way that is useful for both humans and computational tools. We would like you to learn how to integrate genomic heterogeneous data within sound databases, employ genomic databases effectively, and also allow to easily formulate queries to answer complex biological questions. We will start with a motivation for our tutorial, explaining how conceptual models can be used to express, to describe the genome, but also how conceptual models can be used for designing databases that search and compute over the genome. We will do this on human genomics cases, which include the genomic data model, the genomic conceptual model with its integration framework, and the search engine, GenoSurf. We will see genometric query language engine with optimizations and the federated system, and also GECO agent, which is a user-friendly system that overcomes the technological barriers for biologists. Then we will pursue viral genomics with many interesting aspects such as viral characteristic, changes, and spread. Here we will see how viral data has attracted an unprecedented attention. I will give you a very brief crash course on viral biology so that you can understand the following of the tutorial. We will see our viral conceptual model, how it was used to design the VirusSurf search engine for finding sequences and mutation patterns, Episurf to search mutations on epitopes ranges, then VirusViz, a visualization engine for mutation distributions, and ViruClust to compare in time and space the mutation of SARS-CoV-2. Finally, we will put together genotype and phenotype of hosts and viruses and conclude our tutorial. So this is the first part. The second will be presented by Pietro, then I will be back for the third one, and Pietro will be back for the final part. Now, a motivation. Here you see a very famous diagram that shows that around 2008, the cost per human genome suffered a strong reduction. It went from about $10 million to around $1,000 per genome. Well, this decrease in the cost has not been matched by many measures in the organization of these genomes. Here we see a growing number of experimental samples that between 2000 and 2021 went from practically zero, when these gene expression omnibus repositories were created, to over 4, touching the 5 million of samples. And we can say that these are still the ways in which metadata, information about samples in genomics, are collected. You see, this is a spreadsheet for the input of metadata, and it's, well, guided by just text, the explanations. There is no ready-to-fill field or possibility to link these values with ontological terms. So everything is actually collected in a very not curated way. At the same time, in this domain, these are the current ways to model metadata. On the left, you see the one of ENCODE, which is a very important epigenomics portal. And the rectangles you see are the classes of a class diagram or an ER model. And you can see how many relations are used to connect these tenth of classes. Of course, this is probably 1/10 of the whole of the entire diagram. So you can see how people can actually get headaches instead of help from these kind of models. On the right, instead, we have Genomic Data Commons, which is a very important portal for tumor-related data. And here we cannot say that this is actually an ER model, because here we just have nodes for entities, but then it's not clear how they are connected with which semantics. At the same time, here imagine that you have to search for data that is about breast cancer, and I put together the the search interfaces of 3 different data sources: Genome Data Commons, Gene Expression Omnibus, and ENCODE. Well, on the schema level, if you're looking for the information about disease, you may find words such as disease type, primary site, or characteristic and tissue, or health status. On the value level, instead, you may find things like breast invasive carcinoma, breast cancer, ductal carcinoma, mammary gland adenocarcinoma, breast cancer adenocarcinoma. Well, of course, these are all different things, but if one uses support by ontologies, biomedical ontologies, then he may find out that all these concepts are pretty well connected. Some of them are just hyponyms or hypernyms of each other, or synonyms. So for the purpose of a genomic study that has to compare the genomic tracks of these samples, it would be enough to put them together. But this cannot be done because on the portals such linkage information is not available. So a lot of tedious effort has to be done on these external resources. At the same time, when we speak about data in genomics, we have a lot of different signals. So we may have 3D chromatin structure, how the chromatin is accessed, how interactions in the chromatin behave, the story of the methylome, of how the chromatin modifies the transcriptome RNA binding, and a lot of other information. You may observe as different data types have all different schemas. Here, for example, you see you have the chromosome, a start and end position of a region, and then a number of parameters. Here you have the chromosome, some annotation, and then after 3 fields you have the coordinates. Or here, for example, in the third rectangle, the coordinates are not clearly identified. It's probably the 3rd and 6th field, and then there is actually a very long sequence there, and so on and so forth. So you see as different data types are all expressed using different formats. Well, then why do we want to use conceptual models? First of all, we would like to provide a conceptual and also shared clarification of the domain. We would like to be able to simplify and systematize what we call the genomic chaos, building, therefore, efficient data management systems and driving data integration solutions. Finally, for empowering effective search over the genome. Let me start showing you some conceptual models. So, conceptual models of the genome. What do I mean by of the genome? These are conceptual models whose classes or entities directly represent part of the genome. Here, for example, we have Peyton and colleagues that in 2000 represented the genome with its chromosomes, chromosome fragments, which are, for example, connected to genes. Chromosome fragments may be transcribed regions or non-transcribed regions, and these may be regulatory sequences or chromosomal elements, and so on and so forth. Then we have the Biostar models, which had a pretty good success because they were trying to capture different aspects. So here on the top right corner we have the gene sequence. On the top left corner instead we have the patient that has some chromosomes with sequences. Of course, in order to measure the sequences, we perform experiments, and experiments may be of different types, such as, for example, mRNA expression. Here we have instead 2 attempts to capture the structure in 2 dimensions or 3 dimensions of proteins. Here we have the contribution of Ram and Wei of 2004, and then Leon and Pastor in 2021 again started this challenge. Here we have an important project developed at the Polytechnic University of Valencia, guided by Professor Pastor, where they're trying to build a huge and general map of the conceptual model of the genome. Here we have the gene mutation view, the transcription view, the genome view, but many other views are being added since 2016. Now they're probably at 7 or 8 different views, trying to understand all the possible layers and complexities of the genome. And for example, in 2018, they built a new view that represents the variant disease associations. And here I just want to show you that there are conceptual models used also for representing the genome of other species. This is on the left, for example, the conceptual schema of the citrus genome. Here below we have a very simple conceptual model used for a database of bacterial microcompartments. And here on the right, it's schema for just very specific parts of the genome, such as the transderm relational database. And thank you. I pass on the word to Pietro that will continue with the second part of the tutorial.
Hi, everyone. My name is Pietro Pinoli. I'm a researcher in the same group of Anna Bernasconi, and we have been working together on several projects for data-driven computational genomics. Today, I'm here to present you the second part of this tutorial, which is about conceptual models for human genomes, and in particular, how to use this conceptual model to build tools, database tools and services to query the human genome, and in particular, the main experiment on human genomes, such as the next generation sequencing experiments. We use these conceptual models for building tools which have in particular two characteristics. First of all, these tools are accessible, so user-friendly, are easy to use for the users. And the second is performance. Indeed, when we talk about human genome, we often talk about big data, and thus performance really matter. Our line of action has two main parts, which are the modeling of the experiment as a single item in a way that the user can retrieve the experiment that he needs for his research. And the second part is the modeling of the information within each single experiment in such a way that the user can query the information and answer complex biological questions. For what regards the first part, so the modeling of each experiment, we introduced the genomic conceptual model, which is a star schema centered on the concept of item. An item usually is a file, a textual file, which contains genomic regions associated with some particular properties. The item is described by means of four views. The first view is the biological view, which describes the biological process that has been captured by the experiment. Then we have the management view that describes the consortia or the organization that produced the experiment. The biotechnological process by means of which the experiment has been produced is represented in the technological view. And finally, in the extraction view, we have information, internal information, which are useful for organizing the experiment and successfully query the experiments. The genomic conceptual model has driven the creation of an integrated database, which is filled by means of an integration pipeline named Metabase. Metabase has 6 main stages. The first two are the download and the transformation of the data. So the data are downloaded from the sources and usually in a heterogeneous data format and transformed in the simpler key value pairs. Then the key value pairs are cleaned, in particular to reduce redundancies and the application of the data. And then in the mapping stage, these key value pairs are mapped to the entities and the fields of the genomic conceptual model. We then have a phase of enrichment, where by means of external biomedical ontologies, we enrich the terms in the genomic conceptual model. And finally, the pipeline performs some checks on the database for consistency. The transformation phase, of course, is very important. Some sources already provide data in a semi-structural data format, such as JSON, so it is very easy to extract data and then map to the schema, while other data sources, for example, Geo, here in the example, provides data as a natural language description. In this case, we tested sequence-to-sequence model based on transformers to map entities from the natural language description to entities in the geometric conceptual models. In this slide, instead, we describe through an example how the annotation process for the enrichment works. Suppose that we have a biosample about a cancer cell line, a breast cancer cell line, which is MCF7. First of all, this concept is recognized in domain-specific ontologies and linked to the entity. Then each concept is enriched with synonyms at the same level in the ontology. Finally, the concept, when it's possible, is also linked with the ancestor and the descendant in the conceptual, in the ontology, by means of the usual is a and part of relationships. Then, as I mentioned, we also use the model to drive the development of accessible web application. In this slide, we showed Genosurf, which is a web application to browse, to find, to retrieve hundreds of thousands of genomic experiments. The web application of Genosurf is built directly upon the geometric conceptual model and is divided, is split in several panels, each of which corresponds to one of the views of the star schema. The user interacts with the application by selecting the field of interest or optionally using a key value search, so where you can use free text. The result of the query, of a query, is the set of the experiment that correspond to the user query. In this slide, we show the power of the semantic enrichment. Suppose that the initial query is to retrieve experiment, ChipStack experiment from Systrom, and the user is in particular interested in brain. If you just use original, you get 278 samples. If you want to use also synonyms, he gets also other experiments which are connected to concepts which are considered synonyms of brains in the biomedical ontologies. Finally, when the user also selects expanded, he gets much more terms, much more result. In particular, all the results associated with terms which are either ancestor or descendant of brain in the biomedical ontologies. Let's now move on the second part of this section, which is the modeling and the querying of the information within each experiment. So experiments usually come in very different file formats, which depends both on the type of the experiment and on the provider of the experiment. Thus, in order to being able to query all this information together, we need to have a unique data model, which we define the genomic data model. In this model, we have that each region is represented by a tuple, which comprises an ID, which is used to track the provenance of the file, the chromosome, the strand, the start and the stop, which uniquely identify the region on the genome. And finally, an arbitrary set of values, the payload of the region, which contains the information associated with the region. So for example, if a region is a gene, we may have in the payload the name of the gene as well as the expression of the gene. A dataset, sample, in a sample, all the regions have to share the same schema. So must have the same set of attributes. Then regions are associated by the ID with their metadata. The metadata is the same information that we have in GenoSurf and in the Genomic conceptual model. Then, by convenience, samples with the same schema are grouped within datasets. This type of model allows us to perform very complex query on many different kinds of data. So suppose that we have a set of genes, maybe coming from RefSeq, 3 replicates of the same chipsec coming from ENCODE, and a set of mutation, maybe coming from TCGA. We want to perform a very complex query. For example, we want to start selecting the promoter of the gene. Then we want to select those regions in the chipsec where the region is confirmed by at least 2 peaks of chipsec, so these 3 regions. Finally, by a join, identifying the promoters that intersect with at least one of these regions. Count for those promoters how many mutations are intersecting, and finally select only the promoter with at least 1 mutation. We can run this query using GMQL, which is the language we define, geometric query language. This is a query in GMQL, and the query is automatically performed, run on all the genomes, so on thousands of regions. The implementation of GMQL relies on an intermediate representation. An intermediate representation is a directed acyclic graph where each operation has 2 sub-graphs. One graph works on the metadata, and one graph works on the region data. GMQL is then compiled down to intermediate representation, and an instance of the intermediate representation can be executed, interpreted, and executed by an execution engine, for example, Spark, Apache Spark. Having an intermediate representation also allows us to implement different dialects of GMQL. So, for example, we now have PyGMQL, which is a Python version of GMQL, RGMQL, which is a R version of GMQL, and Galaxy and Terra wrapper for GMQL. Galaxy and Terra are 2 workflow system manager. The same way, we can also substitute the execution engine with other engine, for example, Apache Flink, SciDB, which is a array-based database, and a relational database, for example, Postgre. Another perk of the intermediate representation is that at the level of the intermediate representation, we can perform some logical optimization. Some of the optimization we use are the ones which derive from SQL, the normal optimization that we know. Some other are specific for this intermediate representation. In particular, one of them is the meta first. The meta first consists in taking a query. For example, this is a query made of 2 select and a join. Rearrange the graph of the query in such a way that the metadata are executed ahead of the region operation, and the result of the metadata query is used to pre-filter the region data. This allows us on several datasets to reduce the execution time. This is because usually metadata are much smaller than the region data. Then on the execution engine, we can perform other physical optimization. For example, when we talk about Apache Spark, the most complicated operation, of course, is the joining of operation. It is complicated because the join, in this case, join between 2 regions, is a theta join, so a join on a particular condition, that in this case is that the start of one region is smaller than the stop of the other, and vice versa. So if we are on a single machine, we can simply rank, sort this region, and perform a careful scan and get the result. Unfortunately, in a distributed engine such as Spark, the sorting is not guaranteed. And we need new method. For this reason, we define a set of algorithms based on the binding. So in the binding, we split the genome, we partition the genome in bins, and each region is associated to the bins that it overlaps. So for example, this region is associated to the bin 0 and the bin 1. Then we perform the join in such a way that each bin is evaluated by only one executor. And in the case that both the sample from both the region from the first sample and the region of the second sample have regions, we check for overlap. And if at least one of the two regions that are overlapping starts in the bin, the given bin, then we emit an output. In the case like this, where none of the region start in this bin, even if we have an overlap, we do not emit the result. This is very useful because we do not need to perform a further step to remove the duplicates. So we just produce the result that we need. We do not produce duplicates. Of course, the performance depends on the size of the bins and the number of executors, as in shown here, with a curve with this shape. Of course, when you have a short bin, you have a lot of duplication, so a single region is associated to many bins. On the other hand, when you have bins which are too large, you have that inside the same bin, you have many regions, you have to perform many comparisons. So the optimal bin is something between here. The same is true also for the number of executors. When you have too many executors, there is a lot of shuffling of data between executors, and you do not gain performance. We also tested implementation on array-based database, in particular on SciDB, where we represented the region instead of list of region as matrices, where each row corresponds to the start of the region and each column to the end of the region. Then to compute queries, we just look at the regions which are close to the diagonal, because outside we cannot have joins. Another test that we did is to use Apache Flink, which is similar to Apache Spark, as an array-based database. And we got very good performance. So each operation is associated to many samples, which have the same operation with maybe the same schema, of course, but different payloads. To conclude, I would like to present you some application of GMQL that we have developed in the recent year. The first, of course, is PyGMQL, which have two main important characteristics. First of all, allows the user to run a query both on his local machine, so using maybe in-house data, private data, and on the GMQL service, so delivering the computation to external service. The second very important characteristic of GMQL is that the user can run query using GMQL and at a certain point materialize the result, the intermediate result in a a Python data structure on which it can run common operations such as visualization or statistics. Can even modify the data and re-upload the data to the GMQL to continue the query. In this case, we have an interactive execution of the query. The second application is a federated GMQL. Suppose that we have several instances of the GMQL system, so each one with its own repository that are connected together. We design a name node that stores the list of the instances, group of instances, so instances in the same group share the same data, so can share the data between themselves. And finally, the list of all the data with the location where the dataset can be found. A query in federated GMQL, maybe from the user that is sitting at this instance, is written like that, where the user uses as additional parameter this, for example, at Sineca or at local, which identify where this particular command of the query has to be executed. So maybe you can decide to execute some part of the query on the local to preserve privacy of the data and some other part of the query to a large cluster for performance region. Again, we use the intermediate representation, which is now divided across several executors. And at each stage, the data are materialized, moved through the internet to the next executor, executed, and again moved to the last executor. The last application that I'm going to show is a GECO agent. GECO agent allows the user to query GenoSurf to find the experiment, visualize the data, and apply some analysis, some statistical or machine learning analysis. What is the peculiarity of GECO agent is that everything is done by means of a conversational agent. So the user do not interact with the tool that we have seen so far, but with a conversational agent, with a chatbot that asks questions, the user reply and search the data he needs. The application is multimodal, so has both a conversational agent and a panel with visualization. The visualization helps the user gain insights, gain intuition in the data that he is exploring. With this slide, I conclude this part, and I leave the stage to Anna for the next, for the third part of this.
Hi again, welcome back. This is the third part of the tutorial, and here we are going to introduce the conceptual models and databases for searching and computing over the viral genome. So we start in this new world of viral genomics that we were interested in from the beginning of the pandemic of COVID-19. First of all, let me say that there has been a growing, incredibly high interest in the literature of SARS-CoV and SARS-CoV-2. Here you see that this peak in red is the number of PubMed database entries regarding SARS-CoV and SARS-CoV-2, well, compared to other works on HIV in blue and influenza in yellow. So only in 2020 there was a peak of incredible dimension. Meanwhile, there was also an explosion of the genomes. The viral genome of SARS-CoV-2 is about 30,000 bases, and the first was started to be collected at the beginning of 2020, Whereas at the beginning of January 2021, we had about 500,000 of sequences, whereas at the end of 2021, we had almost 6 million, and I can tell you that now, at the beginning of 2022, we have about 10 million sequences. So what to do with all these sequences? Well, first of all, let's try to understand something about RNA viruses. What are RNA viruses? Well, they are made of RNA, which is a ribonucleic acid, the genetic material. They are usually single-stranded. This is a case of SARS-CoV-2, but they also include many others that are quite known, such as common cold, influenza, SARS, MERS, dengue, Ebola, the two hepatitis C and E, polio, and measles. So for now, we forget this part where we have the double helix of that DNA that transcribe into RNA, but we just focus on this part. RNA is a single strand. We have adenine, thymine, cytosine, guanine. Actually, in RNA, instead of thymine, we have uracil, and the translation of RNA into proteins makes amino acids. Why am I telling you this? Because I would like you to understand a little bit about how the SARS-CoV-2 genome is made, because this is at the basis of all the works and systems that we are going to see onwards. So as I said, the genome of SARS-CoV-2 is about 30 kilobases. There is a strong sequence similarity with the SARS-CoV genome. It has 4 structural proteins, the one you see in red, which are the spike, which gives the known crown shape to the virus, the E as envelope, M as membrane, and N as nucleocapsid. And then there are 2 big opening frames that give rise to 16 non-structural proteins. Non-structural because they don't have a structural function, but just other ones. One thing that we are interested in when looking at viruses is their ability to mutate. Mutations can be described as with this signature. So the nucleotide in the reference sequence, the coordinate on the sequence, this is, for example, in the 23,403 position along the sequence of about 30,000, and A has mutated into G. Well, we don't only have mutations at the level of nucleotides, but we also have those at the level of amino acids. Amino acids are created by triplets of nucleotides, as we saw in the previous slide. So for example, if we have a triplet that goes from GAU to GAC, which is aspartic acid, if we change the second nucleotide, so the central one, to a G in one of these 4 forms, then we will translate a glycine, which is a totally new amino acid, and therefore will produce a totally new protein. So the way we encode these variations are the name of the protein, then the reference amino acid, the position on the protein, and the alternative amino acid. Why are we interested more than ever in amino acid changes? Because they alter the protein function. And this effect depends on several things. For example, the actual substitution, because from aspartic acid to glycine, for example, many things change, such as physical and chemical properties. Then we're interested in the actual position where the change happens, because, for example, if it's in the spike, in the receptor binding domain, which is the area that attaches to the human host cells, then the mutation may be more critical. And we are also interested in the co-occurrence with other changes. Now let me introduce the concept of variants, also called lineages. They are characterized by several co-occurring amino acid changes, and the cumulative effect of these characterized amino acid changes gives to variants a significant advantage. For example, you know that alpha and delta have become dominant because they had concurrent changes that were giving this advantage. Usually, variants are observed and studied using phylogenetic analysis, which gives an in-depth understanding of how the SARS-CoV-2 sequences evolve. Here you see this is a very big tree, and this upper blue area is all the sequences that belong or are assigned to the alpha variant. And the distance between all these small circles, which are sequences, represent the difference, the distance between their sequences. To simplify a variant, we say that it has some characterizing amino acids. What are the characterizing amino acid changes? They are those that are present in more than 75% of the lineage sequences. So, for example, this table reports the characterizing amino acid changes of the alpha variant, which are 22, of which 9 on the spike protein. Well, we've seen in the first two parts of the tutorial that we spoke about using models, databases, integration techniques, and search systems for human genomics. Well, here I would like to emphasize the fact that we reuse the same paradigms on a novel domain, the one of virus sequences. At the end, the goal was the same. We want to produce easy-to-use systems for biologists and genomic experts using 3 steps: modeling, integrating, and building repositories of data that are homogeneous in the metadata schemata, semantically annotated with specialized ontologies, and therefore can offer homogenized data, which serves the purpose of the third step, which is search. And here, let me introduce you the first basis for the work that I'm going to present later about viral genomics. The first has been designing a viral conceptual model. The viral conceptual model attempts to express all the information that are available on online open data systems where all the sequences since the beginning of the pandemic started being deposited. The conceptual model is centered on the concept of sequence. The sequence is then described by 4 different perspectives. There is a biological perspective, having the information about the host sample, which is the organism that hosts the virus, having information about the collection date, but also geography, country, region, continent, and possibly also gender and age. Information about the virus, because the virus may not only be SARS-CoV-2, but for example, we also care about SARS-CoV, MERS-CoV, dengue, Ebola, and many others. And viruses have complex taxonomy, so in this entity we can keep that kind of information. Then we have information about the organizational perspective, about databases that store this information. We have the technical perspective about the experiment being conducted with which sequencing technology, assembly method, and coverage. And finally, we have the analytical perspective, which is taking care of the variation or mutation at the nucleotide level, such as the reference and alternative sequence with a position, a length, and a type as insertion, deletion, or substitution. Then we have annotation that may be genes or proteins on which amino acid variants may happen. And nucleotide variants may also have some annotated impact that we encode into effects, putative impact, and impacted gene. This model could be compared with what we saw in the first part presented by Pietro, the genomic conceptual model. In that case, we were trying to capture human genomic samples In that case, sequences of human have more than 3 billion base pairs. So they are too big to compare different individuals, aggregate, compute interesting properties. Here instead we have thousands of bases, so sequences are short. Therefore, we made two different choices. In one sense, the central entity was the item, the unit of computation, the unit of deposition in open data repository. So a file of genomic regions with located by the chromosome strand start and end position on the genome. Instead, in viral genomics, we chose to use a sequence as a central item. The viral conceptual model was used as a basis to design a solid pipeline of data and metadata download, import, and storage. Here you see the sources that we use. We have GISAID, GenBank CogUK, and the Chinese National Microbiology Data Center database. We keep two pipelines separate because GISAID has not exactly an open data policy. You have to sign an agreement to access their data and many agreements to reshare them. So we were asked to keep the pipelines separate and not to join the two databases. But basically we use very similar methods. We imported the data, we constructed the metadata, curated the content with standardization, cleaning, computation of metrics, overlaps management, indexed materialization, and then produced a search system that you will see in the following. I believe in part 4 described by Pietro. And finally, here in the pink part, we see that we also import epitopes. These are short sequences of the virus that are very useful to design vaccines, drugs, serological assays. This is because we want to be able to analyze in which epitopes, which of these parts are affected the most by mutation of upcoming growing variants, so that it's possible to understand if some existing vaccines or drugs are affected. Here we have a very impressive overview of the growth of sequences in the two systems. At the end of 2021, we reached almost 10 million. And here is the database that expands and is inspired by the viral conceptual model, because here you see the central sequence with the metadata about host sample, the virus, the sequencing project, the experiment type. Here we have the part about variants with, of course, some views that had to be built in order to speed up our systems. And then we were able to also to link the information of epitopes that are specific for host and virus pairs. And here we have an important and interesting application of mutations. Actually, mutations are very interesting from the point of view of their effects or impacts. Here we have a very preliminary taxonomy of impacts. They are related to protein stability or flexibility, meaning that when a virus holds up a specific mutation, that mutation could give to the virus an advantage or a disadvantage in terms of making a protein more stable or less stable. Then we have problems about epidemiology, such as viral transmission or infectivity. These are not the same thing. Because viral transmission has to do with the ability of the virus to pass from one host to the other, while infectivity has to do with the ability of the virus to start an infection within one host. Then we have disease severity, the fatality rate. We have also other aspects that have to do with the immunology, such as the sensitivity to convalescent sera, so the sensitivity of the virus to treatments in host, using the serum of people that have already passed the COVID-19 disease, or the monoclonal antibodies, or now we could add the sensitivity to existing vaccines. Or yet again, the binding affinity to the host receptor. This is very important, as you see in this figure, as a particular mutation could give the virus the ability to attach easily, more easily or less easily to the host receptor, which is this enzyme that we have in our host cells. And this can also be embedded in our database view, as you can see that amino acid variants and also nucleotide variants can be connected to information, features, effects, also documented by publications in literature. And another way to represent this with a model is our COV2K knowledge base. You see on the left we have what we call knowledge, because here we have the understanding of variants with their names and characterization in terms of interesting position, interesting mutations. All of these can have effects. Variants can have effects, single changes or group of changes can have effects. Effects are the ones that we saw in the earlier slide, and they have some evidence, which is usually papers, forums, anything that is communicated to the scientific community by means of experimental studies, clinical study, or also experiments in lab, in vitro. The changes can be analyzed by means of their structure. They are part of relevant portions of the virus, and also from the point of view of the residues characteristics, what we call the chemical and physical structure before, such as, for example, hydrophobicity, the polarity, the charge of the molecule. Then we can link all this information to what is actually data. So here we have very much simplified what we have in the viral conceptual model, which is a sequence with the mutations at the nucleotide and amino acid levels, but also the host sample with the location and dates. And here we have the epitopes that are confirmed by assays. These two parts can be interconnected. And here I pass the word to Pietro for the last part of the tutorial. Thank you very much for listening.
So, in this first and last part of our tutorial, we want to show you some of the applications that we have designed and built during in the context of mitigating and studying the pandemic of Sarcopte. Once again, those applications are based upon some models, the various conceptual models, and there are two goals in mind, which are, first, we want to build applications that are accessible, so do not need coding capabilities to be used. You need to use scripts to use the application, but are one application that can be accessed by biologists or virologists. Second, this application needs to cope with large data. Indeed, nowadays, end of March 2022, we have approximately 10 million sequences available for investigation. The first of this application is Biostar, which is similar to Genome, and is an application to query to find sequence in the database of sequences. In the integrated database of sequences, of course. So, the application has a first part, first panel with general description, a second panel where you can query this part of the conceptual model. So you can query on the metadata of the sequence. So, for example, the sequence is very important, the sequence project, so we produce the sequence, the associated publications, so on. Experiment types, so this how the sequence has been produced, and maybe some data about the quality of the sequence, and of course the type of virus and type of host for which the sequence has been taken. Then you have a second panel, actually, where you can query this other part of the conceptual model. So you can query for mutation either on the mutation, so the genes of the virus, so on the amino acid mutation, or on the nucleotide mutation directly. The result, similar to the observed, is a list of sequence indices with associated all the metadata describing the sequence itself. Then we did some study about epitopes. Epitopes are parts of the protein produced by the gene that leads the virus. You can see here in yellow, and are very important because this part of the protein can be recognized by an antibody of the host, so the antibody of, for example, the human body. These data are collected by a large database, IDB, that currently has 1 million epitopes for many virus types, and about 6,000 epitopes for Sarcopte. In particular, most of them are on the spike protein, of course. The epitopes are very important because are used to design vaccines. So, for example, Pfizer Moderna use all the epitopes of spike protein, and other vaccine again use a set of epitopes to produce vaccine that train our immune system to recognize the epitope, so the sequence of the virus. And also it's very important to track mutations that are found on epitopes in the population, because when you have a mutation in the epitope, you are going to change the yellow parts of the protein, so the immune system may not recognize properly the protein. The application we designed for the investigation of epitopes is Episurf. Episurf collects data from the banks of virus and IDB, so the database of epitopes. Merge the two and provide an interface where you can query on sequences, so each part, find a particular population of sequence, and on epitopes. As a result, you can see which epitopes and in which proportion the epitope that you selected are mutated in the population of the sequences that you have selected in the first part of the application. It is associated to a visualization, so for example, we can check for some important, some known epitope changes. For example, change of the epitopes of the receptor binding domain in the delta. So this spike protein, the receptor binding domain, is a part of the spike protein. And you can see that in this population, in the delta variant, there are two very frequent mutations in the receptor binding site. Mutations that are present almost in the 100% of the regions of delta. Another important domain of spike is entity, and is well known to be a domain that can be easily recognized by the immune system, thus if mutated, the immune evasion is easier. It's again a domain here in the spike protein. And again we can see that in data we have a particular epitope here which is very mutated, and another epitope here which actually is not has few mutations. This visualization actually comes from another project which is Biostar, and is a very well design project to visualize data about virus. So population virus. Population can either come from virus source or from some data that the user can provide. So a laboratory can have its own data that are not public and can still use this application to visualize the data. On this application, you can see important populations on the metadata of the sequences. So, for example, how the sequences are distributed in space and time. Report on the sequence, so the number of mutations and if there are similar sequences using BLAST on the genera on virus surface. Then for sequence that you can select, so for example one specific protein or even the full virus genome, you can see the distribution of mutations in that region. And finally, you can subdivide, create groups of mutation divided, for example, on some metadata, such as the time, and see how the distribution of mutation change over the time or over the groups that you define. What is important about these projects that we use it for Sarcopte, of course, but it is easily changeable and can be used for any type of virus. So you can easily change, for example, for Ebola, for Zika, or any virus you want. This is one example of analysis that you can do with virus kits, some visual analysis. So you can take all the sequence from Italians in 2020 and you can divide it in 3 groups. So before summer, during the lockdown period, midsummer, when the restrictions were not present or very limited, and after summer when we have the second wave. So what we can see is that in the first period we have very heterogeneous sequence, so you can see that there were few mutations that were present, while during summer, in particular after summer, the sequence became more heterogeneous. So we have many different mutations that are presented as a portion of the sequence. So the sequences were more heterogeneous. This is because, of course, during summer there were exchange of people going abroad and foreign people coming to Italy, so we imported a lot of variants. One famous case is the mutation variant imported from Spain. So we can divide it on 2 metadata. So before summer, September in spatial metadata. So before summer and after summer in Spain, and summer and after summer in Italy, we can see that in Spain we have these mutations here that were present before summer and also after summer. The same mutations were not present in Italy before summer, but appeared in Italy after summer in a quite large share of the population. Another interesting feature of Biostar is the ability to do comparison over time. So, for example, this is a comparison on data of the Epsilon variant, the California variant, on February of the last year. This should be it, sorry. And we can see that one mutation is a mutation that characterizes these mutations. This mutation that characterizes this variant, the Epsilon variant, goes during the time. So these are periods of 1 month each. You can see that in the first month they were very rare in the population, but after 3 months they start to be very frequent in the population. The next tool we want to present is Biocrest. Biocrest allows you to compare two sets of sequences. One is the target and another is the background for their mutation, particular amino acid mutations. The target and the background can be built according to some metadata, in particular the lineage of to which it belongs, the time of the space. So, for example, we can have that the background is U group and the target is just one country, Italy, for example, or that the target is the month of March and the background is the 3 months before, or any possible combination of these metadata. The example shows an application of Biocrest on data of delta, where we selected as target the delta sequences in India and as background the delta sequences in the rest of Asia. We can see from the spatial analysis, so when we compare India and Asia, that there is a set of mutations, that is very present in the background, so outside India, but much less present in the delta sequence of India. If we move to a temporal analysis, so we compare how the evolution of this sequence in India compared to the evolution of this sequence in Asia, you can see that there are the same mutation that starts to be very present and became quite 1% in outside in Asia, so in the background, while stay very few present in India. They did not grow so much. This one instead also have a growth in India, so we can postulate that maybe this particular mutation provides some advantage to the pipes. So, looking at graphs like this, we can understand if some mutation grows in a particular target, so in a particular subset of mutation, and infer some possible explanation for this growth. Finally, this is a overview of all the projects that we have, actually a part of, because we are working on some others. And so from these sources, which are Genbank, the code UK, ESAID, and the IADB for Epitopes. We built 3 databases. In particular, we have Biostar, Biostar for ESAID, and a version of Biostar for ESAID not in a relational database, but in MongoDB, which is used in Biocrest. We have different data models, the VCM model, and the COV2PA model, and we have a bunch of tools. Some are built both for the resolve on the integration of Genbank and COV2PA. Some other are built on the database before the sequence of ESAID. This is because the sequence of ESAID cannot be merged with other sequences by legal statement. To conclude, this is a novel initiative that so far we have the model but not so many data because it's actually difficult to get this data, but we think that in the future this will be very important, where we can directly connect a patient with all the description of the patient, so comorbidities, demographic exposure to some risk factor, particular actions needed on the patient. So you can have also the genetic profile of the patient with the virus sequence, with again the mutation to the particular type, the particular mutation that the virus has. This is very would be very important because from such a structure we can exploit in depth the relationship between a virus, maybe with his particular changes, so the very specific type of virus, and a patient, again with his genetic profile or his comorbidities or his clinical symptoms, clinical aspects. So, to summarize, we have seen that we have conceptual models, we build conceptual models, we have focused on conceptual model for human and viral genomics, and we show that we can have a conceptual model that describes component, so to provide a descriptor of the knowledge of some domains. As well, we can use this concept as we can use some conceptual model to build application that integrate data, allow to integrate data, and allow to search data. And from conceptual model, it is easy to build data designed for accessibility and also for performance. The simplicity of the conceptual model allows also to have many different tools. So, for example, we have seen for Genome that we have several tools that actually do different stuff with one same model, because the simplicity of the model allows a large flexibility to extract many information with different analysis on the same data. These are the publications regards the genomic projects. We have about the description of the model, the integration, the language, and as was the GPO agent, so the conversation agent, and relatively few, because the project of course is much more recent, about the biogenomics. So the conceptual model again about search and visualize sequence, the works on epitopes, and the works on groups of sequences that are better background. To conclude, we would really like to give a special thank to our collaborators, in particular Professor Ceri and Professor Marco Masseroli. Stefano Ceri was the PI in both the projects, and Marco Masseroli was very active and helped us a lot in the project of human genomics. We want to thank Alex Anatolo, who was a postdoc in both the project. Matteo Chiara is a researcher at the University of Milano and is working with us, providing very good insights for the viral part. And Tommaso, Alfonso, Baccarat, Luca Girasi, and Andrea Pollino, who are a student, PhD student and a master student, that work with us to build the software that presented. Our group actually is much larger. We have a lot of graduate students that with which we have still good relationship. These are me and Arif, who are the senior researchers, the three principal investigators. We have a bunch of PhD students. We got a lot of master students working with us, and we have many collaboration, either with Italy and Milan in particular, but also with Europe and around the world. So this concludes the talk. Thank you for your attention, and we see you in the interactive session. Thank you again.